Yeah, good morning my dear students. Today I am going to discuss about the applications of electrophoresis. The first and foremost important application of electrophoresis is capillary zone electrophoresis. What is the use of capillary zone electrophoresis? It is useful for wide range of ion sizes and particularly useful for small volumes of small organic and inorganic ions. So whenever you are having different type of ions and having the small organic or inorganic ions, we can easily determine with capillary zone electrophoresis, particularly the nanoliter volume of the biological sample. Right? Whenever we are having a very small quantity of the ions like sodium ions, potassium ions present in the renal tubular fluid, if you want to determine them, we can easily go for capillary zone electrophoresis. The second important application is the sieving action in capillary gel electrophoresis. As I told already, capillary gel electrophoresis is a slow phenomena as compared with zone electrophoresis. And here in capillary gel electrophoresis, we will have the sieving action, which is more effective than the differences in electrophoretic mobility in achieving the separations of macromolecules such as oligonucleotides, DNA fragments, and proteins having similar total charges. So this is also one of the important application. You can able to separate whether oligonucleotides or proteins or any type of DNA fragments having similar total charges. We can easily estimate with the help of capillary gel electrophoresis because they have the sieving action, and whatever gel material we are using, they have the selective sieving effect, and molecular weight of the sample can be easily identified. So size and molecular weight plays a crucial role here. The third important application is capillary isoelectric focusing. Isoelectric, the terminology isoelectric focusing means we are able to identify the particular amino acid based upon their isoelectric point, pH values, right? So this is one of the important terminology we are using. So capillary isoelectric focusing we are using. The mixture is separated into very sharply resolved motionless bands. At isoelectric point, the particular amino acid, if an amino acid is having an isoelectric point of 6.2, at that particular point they will not show the movement. So easily we can identify the particular amino acid in that particular isoelectric point, right? So what they are doing, the mixture is separated into very sharply resolved motionless bands whose relative positions corresponds to their isoelectric point values which are characteristic for each compound may help to identify it. So for every compound it is very characteristic. The isoelectric point is very characteristic. So based upon this particular technique of electrophoresis, the name of the technique is capillary isoelectric focusing or CIEF. Using this technique we can able to identify the particular amino acid at their specific isoelectric point. Yeah, the fourth important application is micellar electrokinetic chromatography. We shortly abbreviate this as MEKC. Micellar electrokinetic chromatography. What is the purpose? Here we are using some detergents, right? Sodium dodecyl sulfate we are using as a detergent. So what will happen? They will try to form the micelles, right? So they have the critical micellar concentration. So this micellar electrokinetic chromatography, the technique which is focusing on the introduction of chromatographic separation of neutrals. This is very important. Particularly the introduction of chromatographic separation of the neutral compounds in a capillary electrophoresis system. If you want to see the separation of neutral compounds in the electrophoresis system, obviously the ultimate method is micellar electrokinetic chromatography. So introduction of cyclodextrins. Cyclodextrin, if you are introducing into the gel electrophoresis to particularly this MEKC separation, particularly here in this electrophoresis, if you introduce the cyclodextrin to an MEKC separation, they change the times that enantiomers of optically active compounds spend in each phase. So they will just change or alter the times that enantiomers of optically active compounds spend in each phase and affect the chiral separations. So particularly in chiral chromatography, the stationary phase what we are using is cyclodextrin. There are different forms, plus form, minus form, different optically active enantiomer we are using as a stationary phase. So if you use cyclodextrins here, so what will happen? They can just change the times that enantiomers of optically active compounds spend in each phase and they affect the chiral separation. So this is also one of the important application of electrophoresis. These are all the types of electrophoresis: capillary gel electrophoresis, capillary zone electrophoresis, capillary isoelectric focusing, and then micellar electrokinetic chromatography, right? The fifth application, the very important or the primary application of electrophoresis from long days is for the large biological macromolecule separations. We can able to separate the large biological macromolecules on planar gels. We call them as one-dimensional planar gel electrophoresis. This is the primary application. Since long back we are using this application as one of the important application in electrophoresis. The next important application is many DNA, RNA or protein digest. Right? Many DNA, RNA or protein digest, they contains thousands of fragments or hundreds and thousands of fragments and extracts of particular tissues or cell types. So they have different cell types. From yeast if you are taking, they have different cell types, right? Which may contain similar number of proteins. They may have one type of protein and another type of protein. They have some similarity which you cannot able to resolve by separation in one-dimensional planar gel electrophoresis. So you must go for two-dimensional planar gel electrophoresis to resolve those particular compounds are having similarity, right? So we are using two-dimensional planar gel electrophoresis. This is also one of the important application of your electrophoresis. The next important application is protein peptide mapping. What is this protein peptide mapping? So this can be performed with the two-dimensional planar gel electrophoresis. Again, we can perform the same technique with two-dimensional planar gel electrophoresis, just used to have a counter plot where we can able to map the complete proteins and peptides in a single electrophoresis system slab, right? So here we can use the protein peptide mapping. This is one of the very important technique with the two-dimensional electrophoresis. Now, there is another application. The most common standard protein or peptide mapping experiment employs first a slab gel electrophoretic separation. If you go for the standard protein peptide mapping or protein or peptide mapping, we just go for standard slab gel electrophoretic separation employing isoelectric focusing. Here we have to apply the isoelectric focusing technique also to spread the amphoteric analytes. Amphoteric analytes are having both the characteristics, right? So to spread the amphoteric analytes across one axis of the plate, right? You have to spread the amphoteric analytes across one axis of the plate in order of their isoelectric pH values or isoelectric point pH values. So this is also very, very useful. Particularly if you have the amphoteric analytes, you have to have a proper spreading in protein or peptide mapping, right? So these are the very important applications. Other than that, some other applications are also there for electrophoresis. One of the important thing is that agar gel electrophoresis. If you look at agar gel electrophoresis, it is an ultimate choice in immunoelectrophoresis as such, where antigen antibody are involved. Then we can use starch grain electrophoresis, which is used for the separation and isolation of enzymes, right? Particularly isoenzymes. Isoenzymes are the multiple forms of the enzymes. So in different tissues, they perform the similar function. If you take for example CPK1, CPK2, CPK3, there are different isoenzymes for creatine phosphokinase, right? CPK2 is present in the heart, which is responsible as important biomarker for myocardial infarction. So if you want to separate and isolate the particular isoenzymes, the only method is starch grain electrophoresis. Now electrophoresis is mainly used in the determination of high molecular weight compounds like amino acids, proteins, nucleic acids, etc. The same technique electrophoresis can be used for DNA sequencing also. So one of the example for DNA sequencing is recombinant DNA sequencing. Now the electrophoresis is also used for DNA hybridization. Some platform tools are micro hybridization array technologies we are using. So all these cases we are using electrophoresis for DNA hybridization. The same like your microarray technique. Again, it is used in the determination of biological fluid constituents. Example, blood constituents. If you want to analyze the blood constituents, again you can go for electrophoresis. It is used in antibiotic analysis. Classical example is penicillin determination. We can also use the technique in purification of vaccines. Example, influenza vaccine. Now it is also used in chiral analysis. Example, chiral chromatography. We can use the chiral chromatography also in adjoining with this electrophoresis. Also used in the determination of impurities. If you want to determine the impurities, again we can perform this electrophoresis technique. It is used in some specific plant compounds like atropine phosphate and some codeine assay. Right, synthetic compounds like codeine assay, we can also do this with the electrophoresis chamber. Also useful in diagnosis of hereditary diseases and genetic toxicology. There also we are using the electrophoresis technique. It is used in carbohydrate analysis, like example invert sugar. We can analyze the carbohydrates easily with the help of electrophoresis technique on a slab gel. We can also use the technique in forensic sciences. Example used in the composition of fingerprint analysis. So this is also fingerprint analysis means DNA fingerprinting. So that is classical way we can do with the help of this electrophoresis technique. Then overall applications, if we want to look at that, we can use the technique for diagnosis, separation, isolation and purification in various areas of molecular biology. Wherever DNA, RNA, proteins, all these things are involved, there we are using this as one of the very important unavoidable technique, that is your electrophoresis as such. So if you look at that, one of the example is electrophoregram. This is a graph obtained in capillary gel electrophoresis. So if you go for capillary gel electrophoresis, whatever the graph we are getting, to identify the various nucleotides or fingerprinting, all these things we can analyze with the help of electrophoregram. Electrophoregram is basically a graph which is obtained in electrophoresis, particularly in capillary gel electrophoresis, and that graph will give the complete identification of sequences of DNA molecules present in the sample, right? So this is called electrophoregram. So I hope you can understand in a better manner. Thank you, my dear students. Thank you very much.